Hello, welcome back. Okay, this is the next single cell sequencing method, and we're only going to cover these two. I think these give a nice breadth. There are different ways of doing things now, and yeah, it's a fascinating area. And if that ends up being what you go into, then you're going to have to learn a lot more about it. But for everybody, since I don't know where you're going, let's do a little bit. Let's get you more or less up to speed so you can have a conversation with people if they're doing single cell PCR and you don't feel completely like out of your depth or out of water or whatever. All right. So the next method is called a REAP-seq, which is an abbreviation for RNA expression and protein sequencing. So you might say, well, if we make an RNA, we make a protein, right? Yeah, of course. But sometimes that is not the case, because there can be a lot of things that the cell does to control the amount of protein that is produced after the point where transcription happens. For example, some RNAs are stored up inside the cell, and so they don't actually get translated, even though they get made and build up over time. They're sort of sequestered. They're hidden away for later use. An example of that would be the nucleolus. That's almost, they call it like a liquid crystal. It's so packed, but of ribosomal RNA. Or a thing called a P body, P like the letter P. And these are involved in inflammatory responses and antiviral responses, for example. They'll have a lot of messenger RNAs for things that the cell would need to respond in a hurry. And so then the cell doesn't have to recognize it's infected, send a messenger to the nucleus, start transcription, get an RNA out, splice it, and then translate it. It already has the messenger RNAs ready. It just has to release them, translate them, and then start making defense proteins. It's it's a good thing. It's like getting a step ahead in terms of infection. And there's also regulation. There are small interfering RNAs that we can put in. There are microRNAs that occur naturally in the cell or are made, in some cases, by pathogens. And those can bind to and shut down RNAs, messenger RNAs that have already been made, and they can prevent them from being translated. There are some protein complexes they can call in that'll actually break the RNAs into little pieces and silence them. Yes, the whole field's called RNA silencing. Yeah. So the amount of, well, and let's look at it from the other side. The amount of protein in a cell kind of depends on how long each of those proteins lasts, right? Because if you make one and you break it after 5 seconds, then you make another one. You can be making one every 5 seconds, but it'll look like you only ever have one protein there because you keep breaking them so fast. Yeah. Same thing if you have a protein and it lasts for 20 years. You may only express it once. And then every 20 years you may have to make one more copy of it if that's all you happen to need. So protein levels do not always correspond to RNA levels, and you should not ever assume that they do, although in the past people have made this assumption. And been wrong about at least some things. Not everything, but at least some things. So this technique lets you do single-cell protein analysis and single-cell RNA analysis at the same time, which is pretty darn cool. Yeah. How do we do this? Well, you'll be happy to know the single-cell RNA analysis is exactly REAP-seq. It's just like we covered in the last one. So here's our little hydrogel bead. Here's our oil. We're mixing it together. We got a cell in there with the bead. We're going to burst the cell open. We're going to do reverse transcription because our bead has these primers that have a poly-T tail on them. Which is going to bind to the poly-A, and so we're then going to copy the entire thing, and we're going to have a little barcode right in the middle. That's our little cell BC, cell barcode. The additional thing is that each of these, you're going to add in some antibodies, and the antibodies are going to be labeled, not with a fluorescent group. Oh no. They're going to be labeled with a nucleotide. And the nucleotide, I don't know if we can zoom in. Yeah, here, let's zoom in. Let's zoom the heck in. The nucleotide is going to Show us in the next. Oh, here we go. Yeah. So we're going to have a PCR handle, which once again, here's our handle, here's our barcode. So this is, I guess, a read identifier to tell you which one you're dealing with. And then you get your poly T up here. You're going to put in a separate set of these. And they're going to have a poly A tail. Yeah, do you get what's going to happen? Now you're going to put in these antibodies, and if they bind to the correct protein, and this would work well for surface proteins in particular, and only surface proteins. So you can attach potentially one antibody to every surface protein, and you sort of wash the rest of the antibodies away that didn't bind to anything. And so you've got a cell that's covered in little antibodies. That's neat. Then you're going to take that, and this is going to go in, and now every antibody can donate a little sequence that has a poly A tail, and every bead in here is looking for poly A messenger RNAs, and now also it'll bind to poly A antibody RNAs. Each antibody, it's actually a DNA rather than RNA, but each antibody DNA tag, is going to have its own barcode. So you'll be able to theoretically use 65,000 different antibodies in your assay, which can measure, I think, every human protein. We have something like 20,000 genes, but we express several isoforms of each one, like different splice variants, for example. So I think it's closer to 80,000 or 100,000 total proteins that you would get in a cell. But of those, only a small fraction are going to be available at the cell surface to make this technique work. So yeah, essentially, it's okay. It's not a limit. If it's on the cell surface and you have a very good antibody for it, and you can join a piece of DNA, conjugate a piece of DNA onto that antibody, then you can do this technique. So it's limited, of course, by antibodies very much, because antibodies are expensive, and yeah, you can spend a whole lot of money on a assay like this. But you can learn a lot of things that you cannot learn otherwise. And you can correlate the amount of protein on the surface with the amount of RNA that's being made inside. Yeah. And so that's interesting. And so, yeah, there we go, 65,000 barcodes. And so, yeah, you just send in your antibodies with their own little poly A things. They're just like poly A and a barcode, and then you're done. And you're going to burst the cell, which has all these poly A messenger RNAs. And then the beads don't know. The beads don't care. They're just going to copy both of them. And of course, you're going to have they call it the cell barcode, but really it's the bead barcode. And so you're going to know that, all of these protein and all of these RNAs are coming from the same sample. So how does this work? You're going to get out a bunch of sequences of a messenger RNA of some sort, and you're going to have barcodes attached to those sequences, because we're going to get rid of the actual RNA, but we're going to make a copy of it in DNA, and then we sequence it with the standard Illumina sequencing later on. So you're going to be able to read out all of those. And you're also going to be able to read out all of the little antibody tags, because every time there's an antibody tag there, it's going to have an antibody barcode. And so you're going to have a cell barcode and an antibody barcode, and it's going to be a really short PCR product. And so you can separate those out. You can keep the little ones over here and sequence them, and that'll tell you the proteins. And you can keep the big ones over here. Those will be from the messenger RNAs. And sequence them all, and then you have a little matrix like this that says, okay, gene 1, how many messenger RNAs did we find? This many, this many, this many in these different cells. Great. And how many proteins did we find in each of those? And in their example, they have exactly the same number of proteins as messenger RNAs. So 1-to-1 ratio. I'm pretty sure that never happens, but that's nice anyway. So there you go. So that's REAP-PCR, or REAP-seq rather, an RNA expression and protein sequencing. Okay, and so this is from a paper about this. This is the paper down there. This is where they actually came up with this. And this is where everybody takes their diagrams from on the internet, as far as I can tell. And so if you wanted a more technical language, it's there. If you wanted an even more technical language, it's there in that paper. So yeah, have a look at it. It took me 2 or 3 goes to get my head all the way around this. Try and sketch it out. Watch the video back again. It can work. Here's a little zoom in on these tagged antibodies. So here's the different antibodies. They're the Y-shaped thing. And this wiggly thing attached to the side, this is going to be the DNA that's conjugated to the antibody. And so you have to order these specially, these sort of conjugated antibodies. But they can be made. It's expensive and laborious and somewhat difficult, but it can be done. And so then each of these little things is going to have the main read primer. So that's the thing that they can bind a primer to. It's going to have a poly-A tail of about 25 nucleotides. That's great. And it's going to have an antibody barcode. And you'll have different antibody barcodes that come from different antibodies. And of course, when you copy this with the poly-DT primer, you're going to put the cell barcode or bead barcode onto it. And so, yeah, the reads from the proteins are going to be basically bead barcode and then antibody barcode, and then the read is done. And we'll just copy those alongside the other reads, or we can separate out all of the reverse transcribed complementary DNA, cDNA, and we can, that'll kind of separate most of the protein sequences or reads from most of the RNA reads. So yeah, so we're not directly sequencing protein, we're not directly counting protein, we are indirectly counting protein by using an antibody that binds to the protein, and every time that antibody binds, it ends up stuck to the cell, which means it's going to get next to the bead, which means that this little piece of DNA that we attach to the antibody is going to get to attach to the bead, and it's going to get copied by the reverse transcriptase on the bead. So there you go. That's the idea. And so they're showing you their proprietary primer down here. And so good for them. Good for them. Oh, pardon me. I think that's the end. Yeah, that's the end. All right. So hope that was helpful. And yeah, have fun. Stay safe. Learn this. Yeah. And see you on test day. Bye-bye.